welcome back to the NPTEL lecture series on animal physiology. So, in the last section we talked about the pancreas and we talked about the exocrine secretions of the pancreas. So, today what we will do, we will talk about the endocrine secretion of pancreas. So, in the last class I did the complete anatomy of the pancreas and I showed you the islets of Langerhans, and within islets of Langerhans the alpha cells and the beta cells. So, today we will talk about the different functions, endocrine functions of these and how they regulate the glucose metabolism in our body. Just a mild recap before we proceed further. So, we are into, so just give me 1 sec for inches, okay. Section and this is lecture 5 section on digestive physiology. And we are into the section of pancreas and its endocrine functions. So, in the last class I drew this, so I am redrawing it for you. Just something like this, what we drew. So, these are the duct in the pancreas. And there are these cells sitting like this. Okay. Okay, so these are the acinus, and you have some of these cells which are the alpha cells, and there are a few other cells in close proximity where the structure is more like compact structure like this. So, these are the beta cells, and then you have this is the cross section I am showing there. You have the capillaries which are supplying the blood out there, and this whole structure is called the. So, this is the recap of the pancreas cross section which I drew last time, and I told you that some of these cells are involved in the endocrine function. So, the major hormones which are secreted by pancreas are insulin and glucagon. What they do? So, before I draw the schematics, try to understand. So, whenever we take any food, say for example, you are taking bread or rice or something which is rich in carbohydrates. So, what essentially happens is that carbohydrate is broken down into small sugars. The smallest sugar or the functional sugar which body utilizes is glucose. Okay, it could be glucose, fructose, galactose, mannose, likewise. So then these glucose. So automatically what happens when we are eating? So within the blood, the glucose level shoots up. When the glucose level shoots up, body has to have a mechanism to pull the glucose inside into some storage vessel. Why is it? First of all, you have to understand why there is why it is essential. The body cannot have a lot of glucose in the blood, because if the concentration of glucose goes up, what essentially happens when the urine is formed? See, urine will have lot of glucose, and if urine has lot of glucose in it, then glucose will pull a lot of water molecules from your body along with it. So, you will be urinating at a faster rate and you will be losing a lot of water. In that process, what will happen is that a person will be dehydrated, and if this proceeds further, the person will have serious problem, and this is exactly what happens patients suffering from diabetes. You have heard this word that diabetes is kind of very prevalent, and as a matter of fact, India is the capital. It's considered as the capital of diabetes, because there are way too many diabetes patients. In the years to next couple of decades, this will be a huge challenge for India. So, essentially body needs a mechanism by which it can pull the glucose, those glucose molecule back to some storage area. How body does so? So, whenever you are eating, there are certain signals which are generated by the nervous system, by surrounding cells and everything, that request pancreas to secrete insulin, and insulin promotes absorption of glucose molecule both by the liver as well as other organ, where glucose is converted into glycogen, which is a polymer of glucose and much more stable, which cannot be utilized directly. So, now this is the major mechanism by which glucose is being pulled out from the bloodstream. But now the body is in a starved state, body needs glucose. Suppose you are running for you are coming from a long distance run, or you have done a lot of hard work and you do not have glucose at that point in your bloodstream. So, there is a opposite set of signals which are generated within the body, and those signal leads to the secretion of another hormone which is secreted by the pancreas itself, called glucagon. What glucagon essentially does, it outsmarts the insulin signal and leads to the breakdown of the glycogen which was stored. So, again the blood glucose level goes up till the body utilizes it for all its maintenance requirement. But again insulin is there, it is a check post. So, it is the insulin glucagon check post which regulates the blood sugar level, and if there is a problem in this check post, then this leads to either hypoglycemia, where there is lack of sugar in the body, or hyperglycemia, when there is excess sugar in the body. In the body means in the bloodstream. Okay. So, these are the two extreme situations, and hypoglycemia or hyperglycemia is being regulated. The check post is insulin and glucagon, and the master organ which takes care of it is your pancreas. So, this is the overall summary. So, what I will do now, I will just draw this thing, so that it kind of gets engraved into your understanding, and in future if you want to do an in-depth study of this, then you can go further. Okay. So, coming back, so these are the and of course, we will be highlighting that which cells produce what, okay, what alpha cells does, what beta cells does, and how these things all being regulated. Okay. Coming back to the slides. Okay. So, basically what is essentially happening is insulin could be considered, insulin could be considered as a primary anabolic hormone. So, since I have introduced this term anabolic, what is the meaning of anabolic? There are two words, anabolic or catabolic. Anabolic means when from small pieces you are building something. Say for example, you have individual bricks, you are making a building. That is an anabolic process. Catabolism process is that you break the building. So, when the glycogen is forming from individual glucose moiety, that is called anabolism or anabolic activity, and when glycogen is broken down into individual glucose components, that is called catabolic activity. So, insulin is an anabolic enzyme which leads to the addition of the glucose and likewise to glycogen. So, integration or summation of this leads to the formation of glycogen. So, that is why it is called an anabolic hormone. Okay. And second thing is that it promotes the storage of the glucose, fat, and synthesis of protein. Storage and storage of glucose and fat and synthesis of protein. Okay. So, it is secreted by the, so I showed you that alpha cells and the beta cells. So, it is the beta cells of the Langerhans in the pancreas which are involved in the secretion of the insulin, and this name it has got from a transcription factor, okay, this Langerhans cells. Okay. So, what regulates it? Second, okay. So, first an increase, as I was telling you, increase level of glucose, first thing. Second thing, or increase level glucose or amino acids. Okay. Second thing, there is a parasympathetic signal, and the third thing is hormones released by GI tract, such as cholecystokinin. So, these are the three major factors which leads to the secretion of insulin. But then what insulin essentially does? So, some of the things what insulin does is that it promotes the diffusion of this glucose moieties into different storage areas, maybe in the muscle, the liver, and several other places. So, let us enumerate those. What are the different routes what insulin follows, or what are the different approaches insulin follows to kind of channelize the glucose at different storage units. Okay. Let us enumerate that in the slides. So, the first thing it does is stimulate. So, it is stimulating transport by facilitated diffusion to skeletal muscle, where you need a lot of energy, okay, to adipose tissue, that is in your buttocks and all other places where there is a lot of adipose tissue. And insulin also increase the storage of fat as triglyceride in adipose tissue. So, these are the major function of insulin in terms of how it mobilizes the different individual units like glucose, fatty acids, amino acids likewise. Okay. And eventually, in that whole process, some of these amino acids are even to glucose. Okay. So, these are the overall schematics how the insulin functions secreted by the beta cells of the islets of Langerhans. Then what the alpha cells does? So, those are the cells which are involved in the secretion of glucagon. So, let us enumerate the function of glucagon in the slide now. Okay. So, glucagon, which is basically secreted by the alpha cells of pancreas, and this is in response to the secretion is in response to sympathetic innervation, okay, sympathetic stimulation. So, here I wish to highlight something. If you just remember the previous slide, the insulin secretion was stimulated by the parasympathetic, and glucagon stimulation is influenced by the sympathetic. So, I told you while I was teaching you sympathetic and parasympathetic, they have opposing function. So, look, this is a situation we are on the same organ. Okay. So, if I just put it in terms of so if this is the structure of the pancreas. So, it has two sets of innervations which are coming. One set which is, and the other set is like this. This is how this whole thing is being regulated. So, these are the parasympathetic, which are influenced the beta cells, and these are sympathetic, which are influences the alpha cells. And there is always a dynamic equilibrium between the secretion of alpha and the beta cells, which regulates essentially the insulin and glucagon level, and that essentially actually controls your glucose. Okay. So, one second, that is essentially controlling your glucose level. It is a very tightly controlled regulated mechanism. So, then what is diabetes? Diabetes is a situation when the beta cells of Langerhans either fail to secrete insulin, their capacity to secretion of insulin is compromised, or they gets damaged because of some situation like pancreatitis or something like that. So, that is the situation when your body is continuously is unable to regulate the insulin level, unable to regulate the glucose level, and a lot of glucose is being lost through the urine, and thereby leading to a excess amount of fluid loss from the body, which has its effect. In hindsight, it has its effect on the blood volume, on blood pressure, on dehydration, on every chemical reactions within the body, every biochemical reactions within the body. So, if you see a diabetic patient, there are a lot of complications associated with this whole process, and those complications, I think by this time should be clear to you why those complications arise, because the blood volume or the water volume is being compromised. Body is unable to regulate it. Okay. So, coming back to the slides. Okay. So, this is pretty much is where we are sitting, and if I had to put it in terms of, so there are two terms which may, those of you go through different text will come across. One is called the fuel flux in fed state, and the second is that fuel flux in the food deprived state. So, both of them explains exactly what I was trying to tell you. Fuel flux in the fed state. So, when there is a fuel flux, so in other words, what I was telling you in the beginning, while you take a food, it gets broken down by the digestive system into smaller components, for the case of proteins, and it broken down into amino acids. In the case of big lipids, it gets broken down into fatty acids, cholesterol, and in case of carbohydrate it broken down into, which is the major chunk of our food, actually the bulk of our food, it gets broken down into simple carbohydrates like glucose, galactose, mannose, maltose likewise. Okay. So, that is the situation which we call as a fuel flux, and when there is a fuel flux, there is an increase in the insulin secretion. And then the next situation, when there is a fuel flux in the food deprived state, so that is the time when a glycogen has to be broken down into small pieces. Okay. So, that is when, so what we talked about, so is this situation, fuel flux in the food deprived state is regulated by glucagon, and this one is being regulated by insulin. So, this is basically are the two mechanisms which regulates your body's glucose level all the time, and thereby helping you to maintain the balance. And there are two more terms which is of importance are, which I have already mentioned, is hypoglycemia, and there is another called hyperglycemia. Hyperglycemia is a situation when your glucose level is high in the body, and hypoglycemia is a situation when glucose level is low in the body. So, based on this, the action of insulin and glucagon is regulated. Then how we test all these things in the clinic is something called a glucose tolerance test. What the doctors essentially does is that if there is suspicion, what they will tell you, if they suspect that the glucose metabolism has been compromised in your body, what they will tell you, do two tests. They will tell you, one is after fasting and one is after feeding. So, what they will do, they will ask you that tomorrow morning you come to the clinic or wherever you are giving blood sample. So, they will take a blood sample after the whole night you haven't ate anything, early morning you go and you give a blood sample. So, what the doctor will do, they will analyze the blood glucose level after fasting. This is after fasting. Okay. Then they will ask you, you take food and come back after 2 hours. This is a situation when you are, your body is fairly high in terms of glucose. So, then they will measure the glucose level, and then after some time they will measure again to see whether the glucose level comes back to the original situation or not, where it was. Okay. So, essentially the chart looks like this. If I draw it for you, so it is something like this. If you draw it, so this is the overnight fast. So, if this is in terms of time, in terms of hour, and this is, we are talking about the glucose tolerance test. Okay. So, 1 hour, 2 hour, 3 hour, 4 hour, 5 hour likewise. Okay. So, this is an overnight fast. This is a situation of you haven't ate anything during night, or after dinner you haven't ate anything. So, I am calling that overnight fast. Okay. After the overnight fast, so at this point the blood, it shows like this. Okay. Then as soon as you do a fasting, it goes up like this, and then after 3 hours it starts falling, because this is normal. This is how it is. So, if somebody picks up sample at this point, and at say this point, which is fine. So, I know that it has come down to the basal level, but then what happens in a diabetic patients, how the profile changes. So, now what I will do, I will draw the profile of a person who is having diabetic. So, in the case of diabetic patient, the profile goes like this. it goes like this. First of all, the basal level is higher and then it goes up and it remains for a long time and it cannot come back to its basal level. So, if you look at it, so basal level is somewhere out here for this individual. So, anyway he or she has a higher level and it is still not coming back, whereas, if you look at it in the other situation, it has, say, look at it at this time, still it is maintaining. This is the zone where it is still maintaining, whereas, if you look at it, this is fairly narrow, it is a kind of a tipped. Okay. So, this is a case of, this is a diabetic patient and this is a normal individual, and this glucose tolerance test gives you a feel of what exactly is happening in your body. So, what are our options for a person where he or she suffers from diabetes? One of the option is that if some way or other, using stem cell biology, could we pick up any cells of the body and make islets of Langerhans, beta cells of islets of Langerhans, and put them back into the pancreas, which is a dream approach. Other than that, the only other option which is currently being practiced all over the world, people have to take insulin injection. They carry these insulin pouches with them, and then they they have certain sequence, like before taking food they take an insulin injection, or just after taking food or before something like that, they take the insulin injection to ensure that the glucose molecules are being diffused into what I showed you, diffused into skeletal muscle, into liver and all other places, so that they could be stored. So, till regenerative medicine has a way to develop beta cells of islets of Langerhans, or even alpha cells if somebody has a glucagon problem. So, till that time we are solely dependent on insulin injection. So, that is the state of art currently across the world. So, maybe a time will come when we will have those regenerative medicines where the cells will be able to synthesize. You can put the cells back, which are beta cells, they have the ability to produce insulin. So, this is one aspect. So, from here I will move on to the another major organ which plays a very big supporting role in the digestive system is the liver. In case of liver, part of it we have done, if you recollect, while I was showing you how liver has several functions. One of the major function is that liver helps in eliminating the byproduct of one of the major byproduct of red blood cell degradation. So, I will just take a recap and then I will talk about few other things on. So, if you remember, while we were talking about the red blood cells, within the bone, these red blood cells are formed, and these red blood cells have a life of 120 days. And I told you, after 120 days, what essentially happens is that their oxygen carrying capacity is reduced, and these red blood cells are being engulfed by the white blood cells. And once they are engulfed into the white blood cells, so red blood cells have this hemoglobin having 4 subunit, alpha 1, alpha 2, beta 1, beta 2, and in the center you have a porphyrin ring. In each one of these alpha, alpha, beta, beta, they have a porphyrin ring. Within the porphyrin ring there is an iron, and it is this iron to which the oxygen binds. So, iron has to be retained back by the body. So, the first thing happens after, so this is essentially what happens here. So, these red blood cells come inside this white blood cells, which I am showing in yellow. So, here is a red blood cell inside, and then what happens is this. Okay. So, then this iron Fe2 plus, this iron using different siderophores is brought back, broken down into amino acids, which are again brought back to the bones, whereas the porphyrin ring is transformed into biliverdin, and then this is made into bilirubin, and from here this bilirubin is transported into the organ what we are about to talk is into the liver. So, while actually this reaction of porphyrin ring without, so the first thing which happens is that from porphyrin the iron is being removed, and that process is promoted by a oxidation reaction. Okay. Followed by that there is an enzyme called biliverdin reductase, which leads to the transformation of biliverdin to bilirubin, and then this bilirubin along the blood vessels moves to the liver. In the liver what happens? There are two fates which bilirubin met. Out here in the liver, so liver has this canal like structure from where this bilirubin is essentially transported to the large intestine. So, in the large intestine, bilirubin is formed into urobilinogen, and then this is transformed into urobilin, and this is excreted from the feces. This is one fate, and there is other form of bilirubin, stearicobilinogen, stearicobilin, and then that is being secreted, being excreted to the feces. There is another fate. So, this is what is all happening is what is happening in the, this is all what is happening in the large intestine. So, part of the large intestine, and this is where the liver is standing. Okay. So, this is your liver, and this is the large intestine. Okay. And same by the same route, some of this bilirubin goes to the kidney, and it is secreted via urine. So, this whole process of bilirubin secretion is governed by the liver. So, if the liver ducts have some blockage or their architecture had some problem because of cell damage or something, bilirubin secretion is being compromised, and bilirubin is yellow in color. So, as soon as you remove the iron from the porphyrin and transform it into biliverdin, this is all yellow. So, that is why you see in a jaundice patient, they have yellowing in their fingers and likewise. So, that basically is because the body fails, the liver fails to throw away the bilirubin from the body. So, liver has a lot of role in terms of purifying the toxins from the body, and there is another major secretion which is made by the liver, that is called bile secretion. So, bile has lot of fat emulsifying action, but what essentially bile is that what are the different composition of the bile includes apart from water, it has different electrolytes, then it has, as I have already told you, it has bilirubin, it has steroid hormone, it has salts of bile acid, it has cholesterol, it has lecithin, it has phosphatidylcholine likewise. So, it is a very complex mixture what is secreted by the liver in the form of bile, and this bile has a lot of fat emulsifying actions, and it kind of helps assist in the process of digestion. So, this is briefly about the role played by liver, and then we are into the last tailpiece, which is about the large intestine. So, which is a fairly, it has a much larger. So, if you look at the cross section, it looks like this. This is the large intestine, and in between you see the small intestine, which is embedded in between all the. Okay. So, this large intestine, so this is the large intestine, and here you have the small intestine. So, large intestine is mainly involved in extracting the last bit. It has a very limited absorption function as compared to the small intestine, but it ensures as much as electrolytes and water, which could be conserved by the body, one of the major thing it does. And apart from it, what it does is that it ensures the different these bilirubins and all these things are transformed into urobilinogens and everything, and being excreted in the form of feces from the body. So, that is pretty much close in our digestive physiology. So, we will be doing a little bit more under the heading of metabolism and temperature regulation, a little bit more, which serve tail pieces, which I could not really cover out here. So, but that is give you an overall idea about the role of sympathetic parasympathetic nerves, and the role of pancreas, role of liver, role of a small intestine, endocrine role of pancreas, exocrine role of pancreas, and salivary glands and stomach through the conduit tube of esophagus. Okay. So, I will close in here. So, we will take over after this on the section on metabolism and temperature regulation. There we will just do the rest of the different tail pieces which are left. Thanks a lot.